Hello, ladies and gentlemen, and welcome to today's broadcast on Native MS and IMMS analyses of large proteins and protein complexes by Christopher Mallis. I am Jennifer Woods of LabRoots, and I'll be moderating today's event. Before we begin, I'd like to encourage you to engage with us. You can submit as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right, or use the Ask a Question box on the left. Please welcome Christopher Mallis.
All right. Hello, everyone. My name is Christopher Mallis. I'm a graduate student in the David Russell Research Group at Texas A&M University. And today I'm going to talk a bit about some of the work that we've done with native mass spectrometry and native ion mobility mass spectrometry of large proteins and protein complexes. Really trying to study these proteins in their native folded as well as intact states for the protein complexes. So to start, a little bit of background on mass spectrometry. So mass spectrometry really has three primary components. The first one that we're interested in is ionization. And with ionization, the main ionization method that we use in our lab is electrospray ionization. So with electrospray ionization, you have some electrospray solution, or you have some solution in a spray needle with, in our case, a positive potential for positive ion mode, and it's going to form a Taylor cone towards our inlet to our mass spectrometer under vacuum, which is generally held at a negative potential. And this tends to work very well for positive ion mode as we form droplets. Those droplets undergo Coulombic explosions to form smaller droplets and eventually solvent-free gas phase ions that will enter into the mass spectrometer. Now, on the Agilent sources, the electrospray solution is held at ground, and our inlet to the mass spectrometer is held at a negative potential, but that potential drop across between the electrospray needle and the inlet to the mass spectrometer, as long as that forms a negative potential drop between the two, we can get our ions in. The second component is a mass analyzer for mass analysis. In our lab, that primarily consists of quadrupoles, time of flight, mass analyzers, as well as Orbitraps. And then after mass analysis, we obviously need mass detection. Now, some tandem techniques that we can combine, including mass spectrometry, include tandem mass spectrometry, where for our group, for the Agilent instruments, we use the Agilent 6545 XT Q-TOF mass spectrometer. But we can also hyphenate mass spectrometry with ion mobility spectrometry, which I'll get into a bit later in the talk. And for that, we've been using the Agilent 6560 ion mobility Q-TOF mass spectrometer. So I talked a bit about electrospray ionization here. And there's really two types of electrospray ionization that we use in our lab. The first is going to be direct infusion. So with direct infusion, Agilent's solution to this has been primarily through the jet spray technology, where you have some solution being infused into a nebulizer. We form the droplets at the end of that nebulizer with the help of a heated sheath gas. And then these ions make a right turn into the ion transfer capillary and undergo further desolvation with a heated drying gas in this method. But in our lab, we also use what we refer to as static nanoelectrospray. And with this, rather than flowing at a constant flow rate, we can load an emitter, a glass emitter, in this case, with some sort of solution in the low microliter range of volume. And then we have our custom-made tip holder that we fabricated in-house, as well as a syringe for backing pressure. And this fits neatly into the Agilent capillary holder, which is greatly amenable to the Agilent nanospray source. And once inside this Agilent nanospray source, we have a platinum wire is inserted into the solution, which is grounded and held in proximity to the spray shield, which is held at the negative potential to facilitate formation of the Taylor cone and ionization. Now, there's pros and cons to both of these methods. So for direct infusion, one of the biggest pros is that it's very amenable to online HPLC. which allows it to have both high throughput and relatively high reproducibility from sample to sample. That said, with direct infusion, there's a very high sample consumption relative to static nanospray on the order of microliters per minute. It generally requires a wash between samples to avoid contamination. And depending on the type of direct infusion used, especially with microflow devices, these devices can be very prone to clogging, which can be very difficult or even impossible to resolve, especially with stainless steel capillaries that might be used for direct infusion. Now, with static nanospray, we can drop our sample consumption rate down to microliters per hour by loading in the solution into those emitters. We can spray over several hours on a single emitter. And these are often very amenable to native mass spectrometry because not only can we still use buffers and co-solutes that are very amenable to mass spectrometry, namely ammonium acetate, but we're also forming smaller droplets through which are easier to desolvate and are generally going to form fewer adducts on the final droplet and the final ion. However, some negatives of static nanospray is that it's not quite high throughput yet. It still requires a very manual process of loading the emitter as well as with sample, as well as loading the emitter into whatever its holder device is. Static nanospray also necessitates the use of offline buffer exchange. So we will typically dissolve our samples in water and then undergo a sample exchange to an ammonium acetate containing solution. And it requires a much more hands-on approach for now, although there is work underway to try to make this a more automated process. So some of the work that we've done with static nanospray is really to look at native protein complexes. So again, we're using our custom tip holder with our solution with somewhere between 1 and 5 microliters of sample. And generally with these, we get very good native mass spectra of protein complexes. So here's some data on alcohol dehydrogenase and pyruvate kinase. These are both homotetrameric protein complexes of above 100 kilodaltons in molecular weight. And generally what we see is we see very good agreement between an experimentally measured molecular weight and the theoretical molecular weight based on the accepted sequence of these protein complexes. And we can start to tease out some low relative abundance species in the mass spectrum for both of these. So for alcohol dehydrogenase, we see our charge state distribution of the native homotetrameric complex centered at 27 plus, pyruvate kinase, we're centered at 36 plus. But if we look at the center charge state for alcohol dehydrogenase and expand it, we can start to tease out and really look at some low relative abundance, low molecular weight adducts. Here we're looking at the 27 plus tetramer, and we can do some collisional activation and dissociate that tetramer into a high charge monomer and generally a low charge N-1 mer. So in this case, we're not looking at the trimer, but the trimer is going to exist with residual low charge. But we see this monomer at 18 plus, and we can start to observe separation in the mass domain weight non-specific adducts. And based on the charge state the charge states analyzed, as well as the mass to charge spacing, we can start to pull out some of the changes in molecular weight. So we can see that in our intact tetramer, we see an adduct shift of around 660 daltons. But breaking that up into the monomer, we can start to see that there are several different adducts that we're still working to identify what these are, but different adducts on each monomer that are going to complicate the interrogation of that intact tetramer. We can start to see that there are several different adducts bound to alcohol dehydrogenase. We've also looked at some native membrane protein complexes. Again, this work is on the 6545XT. Primarily, we've looked at the ammonium transport channel from E. coli, also known as AMTB. It's an integral membrane protein complex that exists as a homotrimer. And here we're gonna look at some data for double mutant from the Laganowski group at Texas A&M. And in general, at low collision energy, we see a nice distribution of the intact trimer ions centered at 16 plus. And if we increase our collision energy, we can start to increase the population or increase the relative abundance of the monomer as we start to eject out start to dissociate monomers from the intact trimers. And now if we look more closely at the signal for the trimer and look at the zero charge mass spectrum, we can start to observe these low molecular weight adducts identified as 77 Dalton shifts, up to 3 of them, and then look at the signal for the ejected monomer. We can also see that we have single 77 Dalton shifts in monomeric units. And what we've attributed these to is beta mercaptoethanol adducts are bound to the intact, or one bound to each of the monomer, as BME was added to the expression purification protocol to prevent non-native disulfide formation of this cysteine mutant. And this is a really good example, I think, of being able to identify these low molecular weight adducts. We're looking at 77 Dalton shifts on a protein complex whose intact molecular weight is somewhere around 127 kilodaltons. So we've also looked at the GroEL protein chaperone. It's a protein folding chaperone from E. coli. It exists as a homotetradecamer or a 14-mer made of stacked heptameric rings. So if we look at the crystal structure, we can see that there's nice 7-fold symmetry looking down the barrel, and then nice 2-fold symmetry looking across. Intact in its 14-mer state, it's around 101 kilodaltons as an intact complex with around 550 amino acids per subunit. So it's a very large protein, yet sub-megadalton. And this protein is important because it assists in protein folding in vivo with a GroES sort of lid complex that exists as a 7-mer heptamer. And in general, what the accepted mechanism for their interaction is that you have an unfolded protein, U here, that's going to bind into one of the chambers of GroEL, either the top or the bottom. We're going to bind a GroES lid complex as well as some ATP. Hydrolysis of ATP is going to induce refolding of that unfolded protein to a native protein, and then that native protein as well as the GroES lid will dissociate shortly thereafter. So some native mass spectra of GroEL under low source energies. So this is the capillary exit voltage of around 200 volts. We see very nice arrival or mass to charge distributions of the intact 14-mer, as well as some high molecular weight contaminant ions. Now these native ions are above 10,000 m/Z. So if we actually zoom in on this mass to charge range, we can see very well resolved charge states centered at 74, around 75 plus, and with signals going up to around 12,000 m/Z for the intact complex. The zero charge molecular weight for this complex is around 801 kilodaltons, which again is in good agreement with the theoretical molecular weight of GroEL. And if we increase the capillary exit voltage up to 380 volts, we can start to dissociate out some monomer, dimer, as well as trimer ions. And then all of these ions have very similar molecular weights for the subunits. And these molecular weights agree very well with the theoretical molecular weight of GroEL monomers, which is pretty impressive performance for this instrument. We can also do CID of native GroEL instead of in the source region, as we showed on the previous slide, this time in the collision cell between the quadrupole and the time of flight mass analyzer on the 45XT. And then generally what we see is as we increase the collision energy, we start to see a shift towards lower molecular weight of the intact 14-mer complex, shifting from around 800 and 800,000, 873 down to 800,000, 414. So a loss of about 450 Daltons prior to dissociation. And we showed on the previous slide that we can look at the ejected subunits, but if we look at the ions towards a higher mass to charge, we can also see signals for the remaining complex. So in this case, we see signals for the charge drift 13-mer. So we form a monomer with high charge and a 13-mer left over with low charge. We see that these 13-mer signals are up to around 22,000 m/z. So this is really taking advantage of the high mass to charge capabilities of this instrument. And again, like I said before, that we see that shift towards lower mass prior to dissociation. So from this data, as well as the data shown previously, we can start to put together these sort of molecular formulas where we have a 14-mer with a 75 plus charge as the centroid, and this is going to dissociate into a 13-mer with around 43 plus and a monomer with around 32 plus, based on the CID mechanisms here. One thing to note here is that we still see that low molecular weight, or the low relative abundance, high molecular weight contaminant at around 465 kilodaltons. But with the reduced abundance of all the ions, we start to see a higher relative abundance of this 28-mer. That's around 1.6 megadaltons. And this is about the highest molecular weight ion that we observed on this instrument, and I think it bodes well for some of the intact virus work as well as antibody work to really look at these high molecular weight species in their native intact states. So we also in our lab, so this was the work done on the 6545 XT, but one of the focuses of our group is also in ion mobility spectrometry. So for those that are a bit less familiar with that, ion mobility spectrometry is a very complementary separation to mass to charge separation on mass spectrometers. It fits neatly within the timescales of LC-MS, where you have HPLC, where you have separations on the orders of seconds to minutes, and mass spec, where you have separation on the order of microseconds. And most mobility experiments are in that millisecond range. So it fits very neatly within the duty cycles and the time scans of LC and mass spec. Now, there are several sort of flavors or variations of ion mobility, and almost all of the commercial vendors have really pushed in their own directions in these fields, namely with drift tube, with the Agilent system, traveling wave on the Synap platforms for waters, as well as trapped ion mobility on the TIMS systems for Brooker. Now, some of the fundamentals of ion mobility. So especially for drift tube and for traveling wave ion mobility, you have ions that are pulsed into a region filled with a buffer gas under a weak electric field. And these ions are generally separated as well as their charge, where the size and the shape, how they interact and collide with the buffer gas in this region, and the charge is going to impact how the rack field in the mobility device. And with the combinations, we can start to measure some ion-neutral collision cross-sections to be able to compare within different laboratories, within different instruments, within different mobility devices as well. And some of the fundamental math behind it is that you have some mobility K is equal to the velocity of the ions, or the drift velocity of the ions divided by the electric field, where the electric field is the experimental parameter defined by the delta V, or the change in voltage across the mobility device, as well as the length of that mobility device. And if we rearrange these equations a little bit, we get that the inverse mobility is equal to the drift time times the change in voltage across the drift tube divided by the length of that drift tube squared. And if we take it one step further, we can combine this with what's referred to as a Mason-Schaup equation to calculate ion-neutral collision cross-sections, where you have that the CCS, as the omega term, is dependent on the charge Z of the ions, the number density of the drift gas N, as well as the reduced mass mu between the ion and the drift gas, the temperature, and then that inverse mobility term that was defined previously. And this is one of the fundamentals of drift tube ion mobility is that you're able to calculate these from first principles. And all of the other drift tube or all the other mobility devices, the collision cross sections you get out of those are all calibrated or experimentally determined against those determined by uniform field drift tube devices. And now we can take these collision cross sections that we've determined experimentally, again, calculate them based on your experimental results and compare them between different devices. And we can also compare them to those calculated from X-ray or NMR structures available on the PDB. And with recent advances in cryo-electron microscopy, we've also compared some of these values to those structures as well, although I won't be talking about that in my talk today. So some of the work that we've done recently, looking at ion mobility and mass spec of native alcohol dehydrogenase on the Agilent 6560. We're on the left-hand side, we have our ion source, in this case using, again, our static spray custom tip holder, where the ions are then introduced into a high-pressure ion funnel, followed by a trapping funnel, which will trap and release ions into the drift tube for ion mobility separation, and then they will be refocused from that separation through the rear ion funnel, and then eventually guided into the time of flight mass analyzer. So if we look at mobility spectra of native alcohol dehydrogenase, we can look at our data first starting with the mass spectrum. And from the mass spectrum, we can start to identify charge state distributions, in this case for the monomer, dimer, and tetramer ions, with charge state distribution centered at 11, 17, and 24 plus respectively. But we can also look at this data in a two-dimensional plot of drift time versus mass to charge. And from these, we can start to identify our charge state distributions along these mobility trend lines, where we can start to see our monomer, dimer, and tetramer ions like we saw in our mass spectrum, but we can also start to tease out some other complexes, namely here the pentamer of ADH centered at 28 plus, as well as some other complex of around 55 kilodaltons that we're still working to identify what that complex might be, because it isn't a simple transformation of what we would expect to see from the native tetramer. We've also looked at ion mobility mass spec of C-reactive protein, which is a pentameric protein complex of around 115 kilodaltons. And our native spectra from ammonium acetate solutions, we see a nice distribution of our intact pentamer, as well as some dimers of those pentamers. It's seen as decamer signals. And in general, the subunit molecular weights agree really well with our theoretical molecular weight of the subunit of around 23,047 daltons. And we can start to see in the mobility domain that we actually have two conformers. We have this sort of compact conformer highlighted in green, or in blue rather, and an extended conformer highlighted in red. And we can also look at charge-reduced C-reactive proteins. So we can charge reduce it by adding in a small amount of triethyl ammonium acetate, TEAA, as a charge-reducing reagent. And this shifts the charge state down from a center of 23 to a center of 17 in our charge state distribution. And again, has really good agreement between the theoretical molecular weight of the subunits and the experimentally calculated subunit molecular weights. But again, we see this distribution of these two conformers. And if we compare these conformers along their collision cross-section axis, we can start to look at the relative abundance of that compact conformer relative to the extended conformer, both in our native C-reactive protein mobility spectra, as well as our charge-reduced CRP spectra, where we can see that at low charge, we start to populate that low arrival time, low collision cross-section, or rather small collision cross-section signal. So we've looked at, so far, native collision cross-sections or native ion mobility mass spec data, where we try to keep the internal energy of the ions as low as we possibly can through the sampling. But one thing we can do is we can take this a step further and do what we refer to as collision-induced unfolding. So with collision-induced unfolding, we can study the effects of collisional activation on protein structure and protein complex structure prior to dissociation. We can do this by progressively collisionally activating these biomolecular ions to unfold them prior to their fragmentation or dissociation. So in general, how this looks is you have some sort of a compact drift time or a compact collision cross-section profile. And as you increase the collision voltage, you start to unfold the protein ions in the gas phase. As you unfold them, their size gets larger as their charge largely stays the same. And this is going to lead to a shift in the arrival time or the drift time of these ions as a function of collision voltage. So what we can do is we can plot the arrival time distribution or the drift times as a function of collision voltage into a nice 2D plot where we can convert, or we can rather show these sort of waterfall type plots as a nice plot of drift time or collision cross section as a function of collision voltage to look at these transitions as a function of collision energy. Now, to do collision-induced unfolding on any instrument or any ion mobility mass spectrometry instrument requires an activation step prior to mobility separation. So on the Agilent 6560, there's a collision cell in the instrument, but it's located post-mobility. So you would be unable to do collision-induced unfolding by just changing the voltage applied to the collision cell. So in this case, we've partnered with Agilent, especially Ruan Kurogama in Santa Clara to develop this activation cell at the front of the high pressure ion funnel, where there's a potential drop between the exit of the ion transfer capillary as well as this capillary exit lens to accelerate ions into this region, activate them prior to their transmission into the drift tube. And then we can look at the activation of those ions and the arrival time or drift time distributions as a function of the in-source collision energy. So some of the early work that, some of the preliminary work that I've done recently on this is namely to look at collision-induced unfolding of GroES. So GroES is that heptameric lid complex that interacts with GroEL. And in general, under low source collision energies, we see a nice distribution of that heptameric protein complex centered at a charge state of around 20 plus. And as we start to increase that source collision energy, we start to not only dissociate out and start to form signals for an ejected monomeric complex as we reduce the relative abundance of the intact heptameric complexes, but we also start to unfold these complexes. So we can see that as we increase the collision voltage at a certain point, the protein complex will undergo an unfolding step, which is observed pretty clearly in the 21 plus 2D CIU fingerprint, where we start to see unfolding to the secondary complex at around 65 milliseconds. However, one thing to note and one thing that we need to be paying attention to here is that because we might be dissociating this complex, in this case, we see signal that's isobaric with the 21 plus 7 mer complex of the 3 plus monomer that's formed via dissociation. So this is something to be sort of considerate about in these experiments. So we've also looked at collision-induced unfolding of bovine serum albumin using some of the newer technology that's been developed. And in general, at low collision energies, we see the monomeric BSA complex centered at around 16 plus using our device, as well as some low relative abundance dimer signal. And as we collisionally activate these ions, we can start to unfold them. And especially on the 15 plus, we can start to see these different features populate as we start to unfold the BSA monomers into their unfolded non-native states in the gas phase. So in summary, we've worked on, in our lab and with the projects I've been working on, we've worked on development of native mass spec and IMMS capabilities for large proteins and protein complexes, namely using the high mass modes available on the 45XT as well as the 6560 IM Q-TOF platforms. We worked through integration of static nano electrospray emitters with the Agilent nanospray source design so we can limit our sample consumption as well as really push native mass spectrometry on these different instruments. And then we're currently trying to progress towards collision-induced unfolding of large protein complexes and really evaluating some of the newer technology for CIU on especially the 6560 Q-TOF platform. And with that, I'd like to make my acknowledgements. So first and foremost to Texas A&M and my principal investigator and boss, Dr. David Russell, for really helping to push this project along and facilitate the discussion between our research group and Agilent and push really the next steps of this project, which look really promising. I'd also like to thank Jacob McCabe in our group for helping with the creation and really manufacturing of the static spray tip holders. Dr. Xinyun Zhang, who's now back at PNNL, for helping and collection of that data, especially the AMTB spectra, and also her work on the CIU collaboration with Agilent and with Ruan. I'd like to thank the Art Laganowski's group for their donation of the AMTB samples, and Hayes Rise group at A&M for their donation of the GroEL and GroES complexes. From Agilent Technologies, there's a lot of people to thank, including Shi, who helped us get the 45XT work started and gave us an early visit for that work. Noah Goldberg and Ken Newton have been really helpful with getting myself to understand the design of the source region, especially on the 45XT. Ruan's been very helpful and has been working really hard on the design and implementation of the collision-induced unfolding devices used in the 6560. April Holcomb for her help with the installation of that device. John Sawson and John Felstead have both been really, really helpful. And I'd like to thank them for getting this project sort of off the ground and facilitating the meetings amongst everyone, as well as Lauren for helping to organize the logistics for the symposium. Then lastly, I'd like to thank the NIH for funding. We have a pretty good P41 grant and a resource in native mass spec guided structural biology. It's provided a lot of funding and a lot of collaboration with the members of that grant for discussing and trading ideas for these different projects. And with that, thank you for listening to my talk. If you'd like to reach out to me to either ask questions you didn't think of here, or if you would like a copy of the paper that's been recently published on the work from the 45XT that I showed in the first half of my talk, feel free to reach out. My email is on the screen there. And with that, thank you.
Thank you, Christopher, for your informative presentation. We've received some great questions already. Our first question is, how long is the GroEL CID data being averaged for to produce the spectra shown?
So each of the CID scans here, we're probably somewhere on the order of 30 seconds to 2 minutes. With the Agilent software, it actually makes it pretty easy to do sort of sweeping CIU runs where you can, or sweeping CID runs where you can assign a certain collision voltage or fragmenter voltage for that matter, and then acquire each of those for a minute and then swap between the two, so all of your data is collected in a single data file. But yeah, those are probably somewhere on the order of a minute or two per scan. And then for the CIU data at each collision voltage, we're acquiring on the order of less than 30 seconds. And some of the more recent work is really to try to push that number down so we can get more scans in fewer amount of time or in less amount of time for those experiments.
Great, thank you, Christopher. Our next question. Do you know whether your group and Agilent will be releasing some sort of instrument insert for ion activation for CIU experiments?
Yeah, so we're currently working on evaluating a newer generation, a newer design of that CIU sort of source modification. I believe Agilent's plan is to develop that and launch that as part of the 6560 platform, or the next generation of the IM Q-TOF platform. But in terms of when that might be happening, I'm not sure. But I do believe that is a plan in the future.
Okay, great. Thank you so much, Christopher. As a reminder to our audience, questions we did not have time for today and those submitted during the on-demand period will be addressed via the email address you provided at the time of registration. Thanks again to our speaker, Christopher, and to everyone who participated in this presentation. We hope you enjoy the rest of the event. Oh, well, we may have another guest here.
Sure. There was a question that came up regarding status on the CIU, and Chris is absolutely right. The intent for Agilent is to actually launch that capability probably as we get into early part of next year. Sometime in 2021, I would expect to see that capability coming available. And Chris and Dave Russell and his team have been doing a great job of helping us, as also we've been working with Brandon Rutlow also up at Michigan on the same capability. So both labs have been terrific collaborators and helping us with the CIU and developing that. And then that static nanospray also looks very exciting.
Thank you so much. John, do you want to introduce yourself so the audience knows who you are?
Sure. John Sawson. I'm part of the Americas Field Operation, Director of Strategic Initiatives for the Mass Spec team. And thanks again, Chris, for a great talk. Appreciate you guys doing that and the work you've been doing.
Thanks, John. Thanks for all the help.
Well, thank you so much, Christopher, and thank you, John, as well, for coming in. If we don't have any more questions, then I would like to just reiterate that any questions we didn't have time for today or those submitted during the on-demand period will be addressed via the email address you provided at the time of registration. And thank you again, both to Christopher and to John and all who participated in this presentation. And we hope you enjoy the rest of the event.